Great. Well, thank you, Ryan. So yes, hi, I'm Jessica Rupp. I'm an associate professor in the Department of Plant Pathology at Kansas State. I'm actually the applied wheat pathologist there. And so this is a very fun story about how I got into Kernza. So Ryan asked that I tell a little bit about myself. So my folks are from White House, Ohio, little tiny town in Ohio. My grandpa was a farmer, mostly sugar beet back in the early 40s, 50s time period. My dad was the fourth son. The family joke is, what happens to the fourth son on the farm? He becomes an accountant, so that's what my dad did. He was an accounting professor. That brought us to Pittsburgh, Kansas. So I was raised in guerrilla culture down in Pittsburgh, Kansas. And my entire life, love Kansas. It just does something to my soul. And so I've been very fortunate to do most of my life right here in the state. Got my bachelor's in biochemistry and cellular and molecular biology at Pittsburgh. And then moved to Kansas State, where I got my PhD in plant pathology. And I focused on a couple of wheat viruses, wheat streak mosaic virus and triticum mosaic virus. So after that, I had to go get a big kid job, and so I temporarily moved to Montana State University, where I was a faculty member and an extension plant pathologist. And up there, I quite literally got back to my roots, working on sugar beets again, potatoes, and pulse crops. So I was there during a really fun and unique time in Montana. So when I got there, there was 60,000 acres of pulses across the state. 3 years later, we had over 1 million. And so what that taught me is sometimes we jump into things and we don't do the accessory work to find out like what diseases are present and how do we manage those diseases. And so like, can we get ahead of that? So that lends to my mindset that I'm always trying to look to the future so that when farmers are ready for a product, we have a whole suite of information about it. So, you know, I said Kansas was in my soul, and I absolutely love wheat. So when Kansas State called me back, I said absolutely. There I had the applied wheat pathology lab. I coordinate the plant pathology farm out at Rocky Ford. So what does a wheat pathologist do? The family joke is I'm a serial killer. So a lot of the wheat varieties that come out come through my program. So we test 11 different diseases, and we get wheat from Texas all the way north to North Dakota and Montana. And so we test those different varieties for the breeders, let them know how it's done, send that data back to them, and they choose what they want to move through their programs. The focus of my program is actually a disease called Fusarium head blight. It is the most economically damaging disease that we see in wheat. And it's because this fungus here, Fusarium graminearum, produces a toxin, and that toxin is highly regulated in food and feed. So how did I get into Kernza? Well, sometimes you just strike up a coffee conversation, and that's what happened to me. Catherine Turner from the Land Institute was a guest to our department, and so as part of that seminar series and guest visit, she got to come around and talk with all the labs, and we got to discussing like the incredible power of Kernza. Now I'm going to let James go more into detail, I think, about like what Kernza is, hopefully. But I just want to touch on, it's a perennial grain. It was developed by the Land Institute out of Salina. Grain harvested from intermediate wheatgrass, also called Thinopyrum intermedium, is the trade name Kernza. It's a super close relative to wheat. I'll show you how close in a minute. Grain resembles wheat. It's just a little, more elongated. It's a little shrunken in comparison. But it's a very, very cool crop. And so during that conversation, we started talking about where do wheat breeders get some of their best resistance genes? And the idea is we get a lot of them from ancestral crops or closely related crops. We move them into wheat, and then we get the resistance that we want in a background that will grow and yield. And so just to give you an idea of how close that is, I'm not into taxonomy, but you can see everything's the same right here. We get to the tribe Triticeae, and then we get to a genus. And so I would say that intermediate wheatgrass and wheat are basically cousins. So they're pretty closely related. We can do some things, moving genes around. So we were pretty interested in this. And so there are about 200 diseases that affect wheat across the globe, but really only about 50 of them are economically important for growers. Here's another fun fact. We also estimate that wheat actually loses about 20% of its yield to diseases. Some of those are just hidden. We just don't know that they exist. It might be a root rot we can't see. We might attribute it to other things. Anybody who grows wheat knows sometimes we put a great variety out and we solve one disease, and then it just makes another disease apparent to us when we see it. So we have big yield loss potential. Known disease loss is really estimated around 3%. During an epidemic year, something like wheat streak could take out close to 20%. That's billions of dollars. And so it can be really, really tough. So here's our annual rate. Here's Kernza. Now, intermediate wheatgrass is often considered immune. And this is really different language than like the traditional plant pathologist uses, because we talk a lot about resistance and susceptibility. And there's a couple of things that happen under resistance. You might have, a total resistance where the pathogen just cannot infect a plant. And then we also have, maybe like a minor resistance where we can tell it's infected, but we don't take a big yield hit. And then sometimes you hear that termed maybe tolerance if there aren't resistance genes, but there's a background level of resistance. So immunity is a really interesting case. And so, if Kernza is really immune to these diseases that cause the economic loss, one, it's really good that we could potentially mine Kernza or intermediate wheatgrass for genes to pull in and make better wheat. But it also means we need to both protect our crop of intermediate wheatgrass from diseases that it could pick up from wheat, and also protect our wheat from potential diseases that it could pick up from intermediate wheatgrass. And so when we talk about immunity, is it really immune? And so if I showed you this picture, you'd be like, oh, no, look at all that disease. And yes, that does look not great. And we get samples that look like this all the time. The difference is we cannot necessarily tell to what degree they're affecting a Kernza plant in the field or intermediate wheatgrass, truly. And so we wanted to know what diseases are shared among wheat and intermediate wheatgrass, and are the same types of populations there, because we don't want to be planting a problem and leaving it in the field as a perennial agriculture and then, making real enemies of our neighbors or our uncle or whoever's field is next door, right? So I'm not going to go into a ton of detail, but I think there's enough excitement about this intermediate wheatgrass that we know there's a future for it, and I think the development is really being strategic in how it goes on. And so right now, there's about 4,000 acres in development, about 150 growers. Through the SARE grant that Catherine Turner and I got, we got to have some great civic science projects, and we had growers sending us samples, and we just absolutely couldn't do it without them. So we're absolutely thankful to the growers. We've got about 1,000 acres in Kansas and about 30 growers. So what pathogens are actually present in intermediate wheatgrass? Because my lab specializes in Fusarium head blight, and Fusarium head blight produces that toxin that's regulated in food and feed, we're sort of zeroing in on this one. Fusariums, there's like 30,000 different species of Fusarium out there. And so it's a great interest to know what's going on. We want to know mycotoxin contamination, because if you're going to invest and grow this and you happen to get a disease, there's not a fungicide registered to spray on it, right? And a lot of times you might want to be doing organic or at least low input of chemical, So you're not going to want to do that, so you don't want to get rejected, right? And then, of course, are there other diseases that are present but maybe largely unseen? So we wanted to identify the fungal pathogens that were present in grain samples. Why? Because that's what's going to go into your food product. And so if it looks a little ugly in the field from a couple of diseases, but it's still yielding, I think we're okay with that. If it's a grain sample that's going to cause you to be rejected at the elevator or wherever, processor, we don't want that. We want to identify what several of those diseases actually were, test our mycotoxin level, and then one other key component is we wanted to put together some disease guides, because when you are growing a new crop that there isn't a ton of literature out available, something handy that you can put in the glove box or fits in the front pocket of your shirt was one of our goals. So we really wanted to have some extension guides for growers. So we got samples from all over the United States, up in Choteau, Montana, lots from Kansas and North Dakota and Minnesota. So these came to us as like full head samples. So we were able to dissect heads to get grain with all of their associated features in there. So not just naked seed. We wanted to make sure we were capturing awns and all of that too. And so we went through a process where we sterilized samples. I had a previous master student in the lab, Anusha. She was our Kernza maven in the lab. And so they undergo culture. Our goal is to get a pure culture, because in a head of Kernza or in a head of wheat, we actually have a whole population of fungi. It's not just one thing. And so in order to tease apart what's in there, we have to get these pure cultures. And so we grow from a single canidia into mung bean broth. We dissect those out so we get this pure culture. We did DNA extraction, we did polymerase chain reaction, and we used a fungal database to tell us what fungi were actually in those heads. And so 54 out of 79 samples actually did contain Fusarium species. So initially we thought, oh, that's more than maybe we were thinking there would be. There's a whole bunch of others. These are very ubiquitous fungi, but they're not really economically important fungi. So they tend to be everywhere. I joke with people, some of these things you could get off the bottom of your shoe if we just wiped a piece of bread across it right now. So very ubiquitous in the environment, but definitely this was concerning. The other diseases, Alternaria bipolaris, I'll mention. You might be more familiar with this as just a general leaf spot. In wheat, its sexual stage is the cause of common root rot in wheat. So that might be interesting, right? A disease that can be a root and a leaf disease. And then there's some other toxin information with that. So from there, we said, okay, we need to know exactly what species these are. So Anusha went through more DNA sequencing to figure this out. And we actually got a very different landscape than we do from most wheat heads in Kansas. Here we mostly get a lot of Fusarium graminearum. And here we only are sitting about a third Fusarium graminearum. We got a lot of Fusarium incarnatum equisetae complex, which was pretty interesting. That one is so complicated, even the taxonomists haven't really figured out how to pull those two apart. So that one was a little bit surprising. Another one that we thought were interesting was Fusarium verticillioides. This is often more present in sorghum. So from here, we said, okay, we're definitely looking at a different landscape of fungi that we might find in intermediate wheatgrass. So to dig a little deeper, just within Kansas alone, we have very much that same distribution of about, how many. So we believe then, if this is true across all regions and within our own region, that we have a large variety of fungal species out there. Now, I want to be careful when I say, just because we find it doesn't mean it's going to be an economically important disease. Sometimes they're just there. And if we're thinking about a crop that could be immune to diseases, it might be capable of growing there, but maybe it's not growing very happily, or maybe it's not replicating and spreading to other plants, things of that nature. So then we started looking a little bit about like, how does this go in our other states? And as you can see, we had, the same type of variety from Minnesota. From North Dakota, we really only found Fusarium graminearum. Same in Montana, we actually found Fusarium acuminatum. So an interesting story in North Dakota, primarily with their spring varieties that they're growing, Durham varieties, it's probably expected that those are basically a big inoculation pool for intermediate wheatgrass up there. So seeing primarily graminearum makes sense. Fusarium acuminatum, actually when I was in Montana, we started to see this associated with Fusarium headblight of wheat. So I wasn't incredibly shocked to see that. I was incredibly shocked to see there was nothing else there. So I think we need more samples to tell a better story, but it was interesting. So again, here's that layout of these different things. Why is that important? It's because we care about mycotoxin risk. So the most common that we hear in Fusarium headblight is usually DON, or vomitoxin. So this causes feed refusal. It's regulated in the parts per million range. So there's zearalenone that causes a lot of issues in pigs. There's T2, HT2, DON, DAS, and fumonisins. And so each one of these actually does produce a unique toxin. And so when we're talking about testing grain for safety, we have corresponding tests that go with the potential Fusarium that they may be host to. So graminearum, Asiaticum, and Boothii are all DON toxin. But this Fusarium incarnatum equisetae, which was about a third of the samples, actually produces T2, HT2, and zearalenone. And then another really interesting one is verticillioides, actually produces fumonisins. So at this point, we were a little concerned with what we were seeing. Sent stuff off for testing, so the Land Institute has done a great deal of testing. We have sent stuff to North Dakota, Minnesota, and Virginia Tech to make sure we had a good handle on how much was actually in these samples. And it turns out, when we look at regulatory limits, this is not terribly alarming. Just to point out, this is parts per billion, and these are parts per million. So yeah, we're definitely getting stuff. One thing that I would say is most growers who are going out and looking for diseased heads are sending us a biased sample, right? Because it may not be through the crop. And so after a couple of rounds of this, we said, okay, I think intermediate wheatgrass is somehow controlling this toxin level so that it's not as much of a concern. So this is maybe a really good thing for production. So we're pretty encouraged by seeing some of this data. So just to kind of lean back, that does have interesting ramifications for growing Kernza in your field adjacent to wheat. It has interesting breeding implications for wheat, so we have more things. We could mine intermediate wheatgrass for different positive aspects. So I mentioned we had other diseases that we were looking for, and I also said, love some wheat streak mosaic virus and triticum mosaic virus. There's a new technology called nanopore sequencing. It is a long read sequencing. You basically just get down to your RNA and you put it on a microchip. The whole device is about yay big. They used a lot of this during the Ebola crisis, so it's a very cool little piece of technology. And the next day, it will tell you what all the viruses were in that sample. And actually, tissue that looked totally clean, no disease visible on the leaf, was actually harboring wheat streak mosaic virus, soil-borne wheat mosaic virus, and barley yellow dwarf virus at detectable levels. Now, nanopore is extremely sensitive. When we did ELISA on this, which is the industry standard immunological testing, we could not get it to light up. So that tells us something kind of cool, right? So this is where we come back to immunity. It appears that we really do have immunity, because immunity is exposure to something and being able to still make it through with no effects. So that's pretty cool. We had a lot of leaf spots, and this is interesting to me. We had lots of grower pictures come in with leaf spots. Honestly, leaf spots have been around forever. Tan spot is probably the most prevalent that we see. A lot of times when growers were doing wheat on wheat, we would see lots of tan spot. Anybody from southeast part of the state? Sometimes they are spraying for that a little more aggressively than other parts of the state because it's humid, it's wet. A lot of times we have thicker canopies just by the nature of the growing environment. So they get a lot of these diseases. Bipolaris, the spot blotch, has picked up a toxin from tan spot, so it didn't used to be able to cause any kind of foliar symptom, and now it does. And then of course, Stagnospora nidorum blotch is another one. So we were able to pull all of this off of intermediate wheatgrass. So again, I'm probably talking too long, but intermediate wheatgrass, close relative, numerous ecosystem benefits, undisputable, where the fungus is just growing on top, but not the seed itself. So that's something to think about. The other thing is what toxin are we testing for? We are missing, 2 other classes of dangerous toxin if we're only going to look at DON. And so we need to be thinking to make sure fumonisins remain low and our T2, HT2, and zearalenone is low too. Pathogens do move between cropping systems. So intermediate wheatgrass is in the field for a while. And so it's important to know, how do these stands function over time? Like, does viral titer build up? Can we then infect our annual wheat crop, So the idea is like safeguarding both pathogens or both crops from those pathogens. And then of course we want to do further studies on diverse pathogens, their localization, host pathogen interaction, to try and understand what the disease profile of intermediate wheatgrass means in terms of how do we deploy effective disease management for growers to use. So I mentioned those cards. Hopefully if you're at some events with the Land Institute, there's some of these available to you, and we are probably going to get some more printed, so look for these in the future. They should be online soon as well, as soon as we get the final okay. They're really just designed to be note cards that are about yay big. They're printed on waterproof and fold-proof paper. They're on a key ring, so we can keep adding diseases to them. The front of the card shows you what those diseases may look like. The back of the card will tell you the symptoms, factors that favor development, and some important facts. And these are the cards that we've already have made. To get on this list, you have to go through what we call Koch's postulates. So we get that pathogen from intermediate wheatgrass, we culture it, we ID, we confirm that it is, and then we take it back from the plate and put it back on intermediate wheatgrass, and disease has to develop there. And then we take that, we test it, and we make sure it's the same thing we put on. Once we've done that, then it can get a card and say, yep, this is really a host to that pathogen. I'm going to say just one more thing that we have next. So once you start working with intermediate wheatgrass, Kernza, it's really fun and you don't want to stop. And so that's what happened to me. This is Rocky Ford. This is our research nursery out in Manhattan, Kansas. So if you're ever in Manhattan, give me a holler. I'd love to show you the site. You can come tour this anytime.